So it's simply not true that there are no organizing principles in biology, that we don't have some global ways of thinking about things that inform everything that we do. So we all have Newton, and we have his laws of motion, and they're really important, and they're very helpful. Biologists have Charles Darwin, and he wasn't so bad, right? And everything that we do and think about is informed by this perspective of evolutionary biology. And that's the fundamental difference of a neuroscience that's informed by evolutionary biology and one that isn't. Neuroethology embraces it. And if it does, then say, for instance, Nico Tinbergen. Here's a picture of, in this very wonderful book. It's horribly outdated. I still recommend the first few chapters to you. His book, The Study of Instinct, a Form of Behavior. Here's an animal, a three-spined stickleback fish. He is in this strange position, and if this was a colored picture, you'd see there'd be a red belly here. And he writes, male three-spined stickleback in threat posture in front of a mirror. This activity is innate, dependent on internal motivational factors and external sensory factors. It has an intimidating effect on other males of the same species. Historically, it is displacement, sand digging, changed by ritualization. So there's a bunch of words, but those are real processes. And now you can start to understand the complexity of studying behavior and studying how this particular species, say this particular species, has its particular set of behaviors. And that's the way you need to think about it. Okay, those are some of the constraints that we have to bring to a real study of the neurobiology of learning and memory, the neurobiology of behavior. Okay? So that's a starter. If you do that, you can then understand all sorts of different animal behaviors. So we also have our Lorenz model. This is Lorenz 51. In 1951, Konrad Lorenz and Nico Tinbergen and Carl von Frisch received the Nobel Prize for their work on animal behavior. So here you see little geese that are imprinted to Konrad Lorenz. This is the famous old man walking along and the geese walking behind him. This is signaling of a red-winged blackbird to other birds. This is a baby, a herring gull chick pecking at the red spot of the lower mandible, lower bill of the parent. This is a goose, a gray-lag goose, that brings its egg that's been taken out of its nest, brings its egg back into the nest, making these side-to-side motions because the egg is oblong. Once it starts its behavior, if you take the egg away, it continues the behavior, even though the egg isn't there anymore. That's a fixed action pattern, but it no longer does the side-to-side motions, that sensory, moment-to-moment sensory feedback, which is different than a fixed action pattern. So we can bring structure and logic to the study of behavior if we do it the right way. Okay, so that's a quick primer. So we'll open it up a little bit. And this is just a very, very small part of evolution and ethology. It's my bias, because I tend to pay more attention to acoustic phenomenon. We could try to do all of those and get nowhere, or we can pick one and try to make a little bit of progress. And I'm going to spend some time now telling you about a remarkable animal with a very simple behavior that'll turn out to be very interesting to study. And these are these weakly electric fish and their ability to electrolocate, to explore their environment through electric fields. Okay? Nothing particularly fancy about this animal. You can go to the pet store and buy one. And if you put some electrodes into the water, you can listen to its electric organ discharge. You'll hear about that in a few moments. And you can play games with it. You can do many of the experiments that I described to you just now, the simpler ones. You can just do it there. And they're very beautiful little animals. Okay. So they are weakly electric fish. Weakly electric fish have electroreceptors. They're electroreceptive. And actually, even though fishes evolved, the primitive condition is being electroreceptive, mostly it was lost, and most teleost fish, true fishes, are not electroreceptive. But there are these groups of animals that are electroreceptive. Some of them do not have an electric organ in their tail, and they just passively listen to the electricity in the world around them, in the water around them. And some of them have an electric organ. I'm not talking about the strongly electric fish that zap their prey with the electric organ discharge. I'm talking about the weakly electric fish that produce weak signals that create a field around them, and they sense that field and learn things about their environment from sensing that field. So all these electric fish have low frequency receptors, or so-called ampullary receptors that detect DC and near DC changes in electrical signals, but only the fish with the electric organs have high frequency receptors. The high frequency receptors are evolved to detect the discharge of the electric organ.
What's the difference between low frequencies and the high frequencies?
The frequency.
Well, I mean, like, what's the scale?
You can't fool me. Okay, so low frequency is near DC, and we'll see that there are two types of electric fish. And let me go forward, and I think you'll get the answer. So here's a schematic of what we're going to pay attention to. Here's an animal with an electric organ in its tail. It's discharging. We'll talk about that in just a moment. And it's creating an electric field around it, and it has receptors, those high-frequency receptors, all over its body, more in the head than in the tail, but all over its body. And if you put, say, an inanimate object, any object, near the animal, it will perturb the field lines, and the animal should, in principle, be able to detect the perturbation of the field lines by changes in the field as detected by the receptors along the body surface. Okay, so that's the basic behavior. It's a pretty successful radiation. These are examples of a bunch of fish that are sympatric. They live in the same area, the same ground, a bunch of different species, and they have different types of discharges. And at least to some degree, you can imagine you can individually distinguish the species by the structure of their discharges. But also, of course, there are many individuals of any one species living in the same territory. And the question is, how do they deal with that problem? So there are two types of these animals, those that have pulse-like output. And here's a single pulse and the spectrum of the pulse. And here's another one, another spectrum, and so on, different species. You see the structure of the pulse is different. The frequency content, of course, is different. Some are more like delta functions and have very broad power in the spectra. Others are a bit broader and have more concentrated power at some frequency. And the way they avoid jamming is as follows. They fire their pulses irregularly. And if they detect the pulse of another animal, then they wait a bit of time until they detect the next pulse, and then they fire their pulse. And if there's a collision, then they stop and they wait, and with some random amount of time, they fire again. So those of you that know how wired Ethernet works will understand that these animals solved that problem a long time ago. Okay? So that's for the pulse-type species. There are also wave-type species. So the one we're going to focus on today is that glass knife fish, or Eigenmannia, as it's known in the parlance. And they have continuous signals. The wave of Eigenmannia is somewhat sinusoidal-like. There's no way to avoid crashing there, right? If two animals with about the same frequency get close to each other, they're going to interfere with each other, right? No way to avoid it. And those frequencies of an individual animal can vary from 350 hertz up to 700 hertz, something like that, which is the answer to your question. So the Eigenmannia have evolved what's called a jamming avoidance response. So if you now take, you put the electrodes in your tank at home, and you get the discharge of one particular animal, whatever it is, it'll vary from fish to fish, and say there's an animal at 400 hertz. And you put your 2 electrodes in the tank, and you produce a discharge at 300 hertz, nothing will happen. But then as you start to move the frequency closer to 400 hertz, when you get to about 410, 48, and especially, 390, 392, especially 394 to 398. In other words, from 2 to 6 hertz, you approach the animal from below, the animal will raise the frequency that it's discharging at. And you can keep chasing it for quite a while. Okay, you can actually chase it all the way to the top of its range, and then it'll jump below, and then you can chase it down. Okay, so it's trying to avoid the jamming. That's the jamming avoidance response. And here over 4 periods of 25 seconds, you see that the animal is constantly changing its frequency to avoid the jamming signal that it's being exposed to. So, you know, this is it's a very easy thing to think about. So S1 we'll call the signal of the animal itself, and it'll also always be the larger amplitude because the neighboring fish will create a smaller signal on the target animal than the animal's own electric organ discharge. So this will be some other frequency of lower amplitude, S2. And we're going to, we could then plot S1, say on an oscillograph, fix it on an oscillograph screen, plot that against SS, which is the sum of S1 and S2. And as you can imagine, this is just simple summation of sine waves. This point, S1 will stay fixed. S will rotate around this point, and that's related to the beat frequency, right? So you can plot that in a phase plane where the direction of motion is related to whether S2 is of higher frequency or lower frequency than S1. The size of the circle is related to the amplitude of S2 relative to S1. The rate at which you go around is related to the frequency difference between the two. So, or you can think of the same thing vectorially. So, so you know, it's very simple physics, right? And the behavior is just so simple. All the animal has to do is move away from the jamming signal. Okay, how easy could that, you know, how difficult can that be? You just, it's so obvious what the solution is. You just compare the S signal that you detect on the body surface with the internal S1 signal, right? You do a difference between the two, and from that you calculate what direction you have to go and maybe how fast you have to go away from it. Okay, this is what the Doughty first-year engineering student would propose, and it's a perfectly satisfactory solution.
Do they have any kind of detection of like space-dependent detection? So
yes,
okay,
we'll
so
get to that. We'll get to that. The actual problem is a little more complicated. This is a false thing I'm setting up here because that solution is wrong. It's not how the animal does it. And there's a lot of evidence for why that's wrong, and we'll go through a little bit of it in the next few slides. But the fundamental reason is what we as engineers would want to propose is that the output of the electric organ or the output of the neural signal going to the electric organ projects back into the brain and gives access to the actual signal on a moment-by-moment basis. Well, it turns out that the vertebrate body plan did not provide for such a projection. It doesn't exist. And the animal doesn't have actually access to its own EOD discharge in the way we're thinking about it, and so we can't solve the problem that way. So one thing you could do is go, that's boring, it's dumb. Or you could dig a bit deeper into it and discover that the solution that the animal has is extraordinarily elegant, and they have remarkable behavior. They have their champion species at some of the things they do. Their sensitivity is such you can drop a 9-volt battery across the Pacific Ocean, and if there were these weakly electric fish in there, they could detect that change in principle. The discharge of the electric organ is regulated to over periods of, say, 10 or 20 seconds to variance on the order of 100 microseconds, excuse me, nanoseconds, less than 1 microsecond, 100 microseconds, which is pretty good for biological material, right? It's better than your benchtop oscillator. Well, maybe nowadays it's no longer true, but it certainly used to be better than the benchtop oscillator. And they do a lot of remarkable things. So let's dig into it a bit. Jamming avoidance response. Yeah.
So you asked top up or down.
Right.
2 steps. So assuming like the limit cycle, right? Up and down state. So from the attractor, the limit cycle, so oscillating, right?
So this isn't, we're not at the point with anything that we need to think about limit cycles. We're just adding 2 sine waves. Just adding 2 sine waves and getting the beat frequency. I could even do a demonstration with a second person here, right? And we're just looking at how the amplitudes change over a longer period of time than the individual peaks of the sine wave, and how the phase changes between the two. It's not limit cycle, it's just simple summation of some.